All right, thanks very much. I'm going to tell you about the analytical research that we do in the Owens Group, and we're basically a group of mass spectrometrists. And the technique that we work on is a technique called matrix-assisted laser desorption ionization, or MALDI for short. And if you look at all the kinds of things that we do, we really focus on understanding the technique so that we can help this work better. And so we look at biological polymers like peptides, proteins. We do a lot of work on synthetic polymers. And again, it's all using this matrix-assisted laser desorption technique coupled with time-of-flight mass spectrometry. And the two things we really have spent a lot of time over the years working on is the development of sample preparation techniques, and specifically to aim for doing quantitative analysis. When MALDI was first developed, it was really looked at as a qualitative technique, and there were even people who said that it was not a quantitative technique. And we really worked on trying to make it into one, and I think today people have agreed that you can do quantitative analysis by MALDI, although you have to be careful about how you do it. And then certainly we want to develop improved matrix deposition techniques, and I'll show you some of the reasons for that today, in that we could use it for this MALDI imaging technique. Over the years, we've done some laser spectroscopic work with David Miller, Nick Szymanski, and MEM. I'm not going to talk about that today. And you know, when we do a lot of this mass spec work, we develop an awful lot of data, and so we have to work on data processing techniques. And so there's an awful lot of good stuff that's come out of this work. All of this stuff that we're doing, we're working on mass spectrometry equipment, but if you really want to understand a technique, you can't just use that technique to study it. So we're also looking at, you know, a lot of microscopy techniques. We're looking at auxiliary techniques like capillary GCMS, UV visible, FTIR, and fluorescence to try to help us understand this MALDI technique. And so, you know, the question that's probably burning through your mind is what's new and cool in the world of MALDI mass spec on Stratton 4th? I know you're all wondering, right? So what we're doing lately, Brian is leading a look at doing some MALDI imaging work, and Elsa is leading the effort on application of the MALDI technique to improving the identification of microorganisms. So let me tell you a little bit about those things. And so neither of these things is new. Probably the leader in the area of MALDI imaging is Richard Caprioli. And so this is out of a review paper. He talks about taking some kind of a tissue sample. You put the matrix on the sample, and actually that's one of the biggest problems in this whole technique is how you put the matrix down and what effect it has on what you eventually see. All right, eventually after you put the matrix down, you're going to fire the laser at the surface. And what you're going to do is you're effectively going to raster the laser around and take a whole series of mass spectra at all the different locations around the surface. And then with a big enough computer and the right data system, you could take all of this information and put them together into pictures of what the surface looked like. And you find that, you know, this is a very widely used technique now, although, you know, it's in the area of mass spectrometry, one of the biggest growing techniques. And so here's just one example. This would be of whole body imaging. So this was a mouse pup that was sectioned. And so up here you see the optical picture that was stained to try to, you know, visualize what was going on. They took another slice and they put the matrix down, and so you can kind of see all the dots of the matrix that have been deposited on this. And then when they put it into the mass spectrometer and imaged it, what they were able to do then was go back through the data system and produce an image that effectively all of these things were proteins that were observed. And in this particular paper, they didn't even identify all of these proteins, but what they were able to do is to see that they were localized to different locations that could be visualized, you know, in the optical microscope picture. And as you look at this and you say, wow, this is really horrible resolution, you know, we're talking about a technique that from the first time that it was done is only about 10 years ago, and talk about how long it's been since the optical microscopy has developed. And even in the last couple of years, there's been tremendous improvement in the ability to get higher resolution optic or higher spatial resolution than even this. Another place you could do this is looking at samples that came from biopsies. So this is just an example. This was lung tissue, lung cancer tissues. And so what was being done was to look for particular peptides. And these are two biomarkers that are looked at in lung cancer tissues. And what was found in this experiment was that these were from two different patients. And one patient showed one biomarker, right? One patient showed one biomarker lighting up in the MALDI image, whereas the other patient showed both of the biomarkers lining up. And so what was compared to the histology results that the pathologist could say both of these were lung cancer tissues, but on the molecular level, these lung cancer tissues are actually different because they're showing different types of proteins that show up. So what is it that we're doing? What is it that we're trying to improve? All right, so this is just a few, kind of cute examples of things that we're doing, imaging upstairs. So these are done by Brian. We basically we've made samples. All right, we don't have lung cancer tissues, and we don't have, we're not cutting apart mice. And so the idea is that we made some pictures on the surface by stamping compounds of different materials. And so these are different peptides that have been stamped, and so you see in the different colors, substance P, you've got angiotensin 1, angiotensin 2, and bradykinin. They were stamped on the surface, and then matrix was applied on top of them, just as if this was an experiment where this sample happened to come into us, and then it was imaged in the MALDI instrument. And what you can see is a fairly good representation of the shapes that these were stamped in. These were just stamped as, rubber stamps that have different letters on them. And so that's done with 300 microns raster size. And so you see the bar here. This is a 5 millimeter scale, so it's a fairly large image. Here's another example with a slightly smaller raster size, a slightly smaller area imaged. And again, what is it that we're trying to do? When you put the matrix down, how you put the matrix down will affect the signal intensities, and particularly which matrix you put down, it will affect which types of peptides show up better than others. And there's not a lot of information known about this. And so when people are doing imaging right now, they're just putting a matrix down and they're saying, ooh, look at the cool picture. But the idea is that you probably want to have a little bit more information about what's actually there as opposed to just what you see is there. And by understanding the interaction of the matrix with the peptides, we're going to get a better idea of that. And one of the interesting things that we found was this is the picture that we produce when you're actually looking at the ions that are characteristic of these particular peptides. But it's interesting, if you look at the ions that are representative of the matrix, you can kind of see almost, the negative of that. And so where it's dark is you're not seeing as many matrix ions come off the surface, right, as it is where there's no analyte put down right there. And so this is interesting because in a way you kind of could think of it as the absence of matrix is actually also giving you information, the absence of matrix ions. Now in terms of the other project in doing bacterial identification, this was work that came out of the 1990s. Catherine Fenslau and a number of other people were working with the Army to develop biological warfare agent detection. And so as they did this initial work, what was found very early on is that the peptide signal that came out of bacteria samples are actually different and very different depending upon what the identity of the bacterium is. And so this is great. You can do biological warfare detection, but it's probably much more useful to apply this in a clinical setting. And so even very early after that work was reported, people started looking at, could you do this in a clinical application? And so I'm really happy to say that as of last August, finally the Food and Drug Administration here in the United States has approved this method based upon MALDI analysis, and particularly it was pioneered by Bruker, which is the company that did make the instrument that we have. And so what you're now able to do is that if you're sick in the hospital, they can take a sample, and instead of just running it down the lab and doing the traditional tests for what the bacteria are, you can actually do these MALDI tests to figure out what's going on. So again, what's being done here is that they're getting effectively all of the proteins out of the sample. And so this is just a mass spectrum, not taken with particularly high resolution. And what you have is these protein patterns. What they were showing in this particular diagram was how reproducible it was from colony to colony for a single type of bacteria. And there's a scoring system that the company has come up with to try to score these samples. And then these samples are run against a library to see what the identification of the proteins are or what the bacteria are. And so the real object of this paper was to effectively show that by the conventional method and the other traditional method, the 16S RNA method, that this new kid on the block, the MALDI technique, is actually getting the right answers. And one of the things that they were able to find is that it got the answers correct pretty much all the time as long as that bacterium was already in the library. And of course, when they found that there wasn't an issue in the library or an entry in the library, one was added. And so this is now really, really expanding the ability of MALDI to, be used in the clinical setting, to figure out what's going on. Now, we don't just do mass spectrometry in the group. Well, we do a lot of mass spectrometry, but over the years, as people have come into the group to do undergraduate research, they've sometimes said, I'm interested in slightly different stuff than this MALDI thing that you're talking about. And so I just have a list of different things that have been done in the group over the last, few years to give you an idea for the undergrads in the audience. And so this year, Joseph Trusello is working on a forensic application of looking at arson accelerants, particularly gasoline by GCMS. And what's really been interesting to me anyway is how similar all the different brands of gasoline are when you look at them just put them in and separate them and look at them as a complete mass spectrum. But when you go in and you start looking and picking out specific chemical compounds by looking at certain ions selectively, you can then take them apart and figure out, which ones are really which. We've also, again, done some interesting things with, again, investigating the MALDI experiment. And I put this one here, Francisco Guevara, if you know him last year. Actually, this led right to the job that he got at the end of the year. He was doing a lot of work on our Bruker instrument, and he's actually been hired on to be a Bruker repair person, service person. A couple of years ago, Marissa Egan, she's now working at Victory Brewing, and said, hey, could we do this interesting experiment by analyzing beer? And I thought that was a great idea. And so what we were looking for was diacetyl. What is that?
Butter.
Very good. Makes things taste and smell like butter, right? It's not really great in your beer, though. So it turns out that there's a lot of effort to try to understand, how quickly it's formed naturally by the yeast in the beer. But as you let the beer brew, basically the yeast then use it back up. And what you want to do is you want to brew the beer long enough so that it gets rid of that. And so measuring this is usually done by just taste testing, having an expert panel do that. It would be really nice from a little bit more scientific point of view if you could actually follow the reactions a little bit better. You could actually make even predictions and go through and, maybe up the brewing rate of how much gets brewed because you can cut down on the time that you need to let the beer brew. So that's been a neat project that's been going on. Again, we've been doing some other mass spec investigations. And again, there was another person, Alex Belinsky, a few years ago. Rather than look at diacetyl, we were looking at sulfur compounds in beer. I don't know, undergrads seem to keep coming to me saying they want to do beer analysis, and I think that's okay. So we were actually doing that jointly with Phil Handel in the Food and Culinary Sciences Department. And then a little bit longer ago, some of you might remember Adria Wilson. She was doing a project this was an optical project. We were working with a small company. This is a graduate from our department a number of years ago, and they're developing thick film gelatin-based substrates for making basically optical holograms where they could do wavelength separation with these optical elements. And we were trying to study this gelatin material that they were using as their optical material. So there's a lot of stuff that you can do. If you want, come join us. If you want to talk to people in the group, if you want to find me, very easy to find me, either by email or by website.